Okay, this is concept 2 notes, and we're going to talk more about your senses. We left off in our concept 1 notes talking about the sensory division of the peripheral nervous system that takes in your sensory information. And so today we're going to talk more about the organs that are specialized specifically for doing that. So, sensing is just your sensory cells translating stimuli, whether those stimuli are chemical, electromagnetic, mechanical, regardless. It's going to take those and it's going to convert them or translate them into action potentials, nerve signals that our nervous system can receive and integrate and process. General sensory receptors in our bodies are typically modified nerve endings of sensory neurons. Touch is considered just a general or somatic sense, and it relates to our ability to detect pressure, pain, temperature, and tension just through a variety of these general sensory receptors. Here you can see just a cross section of your skin and all the different types of receptors that can be in there, which we'll talk more about when we get to the integumentary system. But there's also just a great 10-minute video that I've linked in the notes section of this slide, just to review the peripheral nervous system to kind of connect these. And it goes into a lot of detail about our sense of touch and pain. But for the sake of these notes, we're actually not going to talk about general senses like touch. We're just going to talk about our special senses. So this is vision, smell, taste, hearing, and equilibrium. These special senses utilize special sensory receptor cells in our sensory organs, like our ears and our eyes, and epithelial structures, like your taste buds and your olfactory epithelium that are in your head. And we're going to kind of walk through each of these special senses now for the rest of the notes. And while we're doing this, my students, I want you to have your big body diagram for the sensory organs out, because you're going to be labeling the structures on your big body diagram and then taking notes just on your Cornell note outline. All right, so let's talk about vision. This is the dominant sense of humans. Nearly 70% of our sensory receptors in our body are actually in our eyes. So hopefully you remember from physical science that light is electromagnetic waves, and we have photoreceptors in our eyes, which your eyes are your vision sensory organ, that are going to convert that light or that electromagnetic energy into electrical energy or action potentials that then travel to the brain in order to be integrated. So let's talk about the vision sensory organ, which is your eye. It has a crazy interesting structure. So even just on the outside, you have protective fat and then the bony orbits in your skull, which we talked about in the skeletal system, just to protect your eyes. And then we have these accessory structures as well. So your eyebrows, they actually are there to help keep sweat and sunlight out of your eyes, because the way they protrude on that orbit there kind of gives you a little bit of shade. Your eyelids and eyelashes trigger reflexive blinking to keep your eyes nice and moist. And then you have this lacrimal apparatus. It consists, it's what's going on in here, of the lacrimal gland that produces and secretes tears, and then some ducts that drain the secretion. So this would be an exocrine gland. So we're not going to talk about it in concept 3. We're only going to talk about endocrine glands, but this is an important part of your eye as well. Another thing on the outside are those 6 extrinsic eye muscles on each eyeball that control your eye's movement, which honor students, we talked about some of the nerves that control these muscles at the end of concept 1. But you got the superior rectus, which elevates and turns the eye medially. You've got the inferior rectus, which depresses and turns the eye medially. The lateral rectus, which controls lateral movements, medial rectus, which controls medial movements, the superior oblique, which depresses and turns it laterally, and then the inferior oblique, which elevates and turns it laterally. So those are the different muscles that control your eye. Now let's talk about what's going on inside. Internally, your eye is basically hollow and it's filled with fluids called humors, which we'll talk about in a couple slides, that basically hold the eye's shape. But the wall of the eyeball, as you can see here, has 3 layers. You can see those 3 different colors here. The outermost layer is the fibrous layer. Okay, this is mainly sclera, which is the white stuff. So when you're looking at your eyeball and you see the white stuff, that's sclera, which is just the anchoring site for those extrinsic eye muscles we saw in this picture. Then we also have your cornea, which is the window that lets light into the eye. So it's just the outermost part of the eyeball's layer is considered the cornea. The vascular layer is the middle layer. Okay, so that's kind of, I don't know if you would say that's peach reddish here in my picture. So the vascular layer has your choroid, which supplies all of the other layers with blood, which is really important. It has your intrinsic eye muscles, which do things inside your inside your eye, like your ciliary body, which is the ring of muscle tissue we can see here around the lens, and your iris, or the colored part of your eye. Your iris is actually just a ring of smooth muscle tissue between the cornea and lens, and it contracts and expands to control the size of the pupil. So the pupil allows your light into your eye. So the iris is essentially, because it's controlling the pupil, it's controlling the amount of light that's coming into the eye. So it's not just there to say that you can say you have pretty blue eyes or pretty brown eyes or whatever. It has important parts and a really important muscle. There's our pupil. Now, these little lines here, you might not be able to see as clearly on your diagram, but these are suspensory ligaments, or also referred to as the ciliary zone. Ciliary zonule. Wow, that was a tongue twister for me. It's just a halo of fibers that are encircling and holding the lens up, because you can kind of see it looks like it's floating here. All right, now the inner layer of the eye, that's your retina. And your retina has 2 main layers. It has the outer pigmented layer, and this has pigment cells that help absorb light so that it doesn't scatter, because we really want to focus the light as it comes in. And then the inner neural layer, which has tons of neurons and neuroglia to support those neurons to help create pathways for the light as it comes in. So, issues with myopia, which is nearsightedness, and hyperopia, which is farsightedness, have to do with how the light gets focused either in front of the retina or behind it rather than on it. And so you can, if you have to wear contacts or glasses, that's because we're having issues with that. A lot of people like to compare the retina to a camera. But there's a good 5-minute video in the notes section here that kind of explains the differences. And also it shows you a couple optical illusions, which is kind of fascinating to see how those work in your eye. So a little bit more about that, your retina. Your retina contains millions of photoreceptors. And those photoreceptors convert light energy into electrical energy, or action potentials, that then travel through that optic nerve we talked about at the end of concept 1 to the thalamus in your brain, and then from there to the visual cortex in your brain to be integrated and processed. So this is that optic nerve. It's going to go out through there. We have 2 types of photoreceptors, 2 main types. Your rods, which register black and white, and then your cones, which are responsible for detecting fine detail and color. And then 2 other structures I want to point out in the retina. The optic disc right here is where the optic nerve just exits the eye. And also we have the fovea centralis, which is that little kind of like bump looking structure right there, which gives us our sharpest and clearest vision. Oh, a fun fact about the optic disc is it's also known as our blind spot because there are no photoreceptors right here where the optic nerve exits the eye. So that's when we say your blind spot, that's what we're referring to. Also in this picture, you can see these red and blue things. What are those? That's just the central artery and vein that are bringing blood and nourishment to the eye and removing waste from it. So there, you can see them there where the optic nerve is. All right. Inside of the eyes, kind of like in the middle, what's going on here? You have your lens, so it's not part of the fibrous layer, or excuse me, not part of one of those three layers. It's considered part of the inside. And the lens is just a convex, transparent disc that focuses that light that's allowed in, so and then it can be projected on the retina in our inner layer. There is, I talked about humors or fluids before. There's the vitreous and the aqueous. So the vitreous is the clear gel that fills the posterior segment of the eye behind the lens. And then the aqueous humor is the clear fluid that fills the anterior segment in front of the lens. Your vitreous humor, fun fact, forms in the embryo and lasts for your entire lifetime. And it helps transmit light and just support the posterior surface of the lens and keep pressure within the eye. Whereas your aqueous humor is forming and draining continuously. And it helps maintain pressure and support within the eye, as well as supply nutrients and oxygen to your lens and your cornea out here. All right, that's your eye. Pretty complex structure, but luckily we're not going to get into too much of the complexity of the structures for your chemical senses. So we'll take a little break there. So your two chemical senses are smell or olfaction and taste, which is gustation. And these are considered chemical receptive senses because they use chemoreceptors to detect molecules in the air that get dissolved in fluids from our nasal membranes when they come through the nose, and then molecules in our food, which get dissolved in saliva. Fun fact, our chemoreceptors are actually sharpest at birth, which is why newborn babies can smell their mom's breast milk from across the room, which is kind of wild. All right, let's talk about smell or olfaction first. What's an odor? It's just the gaseous molecules that we smell. Those molecules hit the olfactory epithelium in the roof of the nasal cavity, which you can kind of see here coming through your nostrils there. And there's a ton of olfactory sensory neurons there that look like this. Your nose hairs are really important, not just a nuisance. They help filter out some of those molecules. Fun fact, dogs have 4 times more of these olfactory sensory neurons than we do, which is why their sense of smell is so much greater than ours. And each olfactory neuron has receptors for only one kind of cell. So we utilize 40 million different olfactory receptors to identify upwards of like 10,000 different odors, which is kind of crazy. So it's a highly specialized special sense. Essentially, what goes down, those molecules come in, they're dissolved in fluids in your nasal cavity. They bind to receptors, chemoreceptors specifically, and then those will then send action potentials down the olfactory nerve into the ethmoid bone, and then eventually to the olfactory bulb that connects to your brain. And so it's traveling down something called the olfactory tract to the olfactory cortex. And one thing that's super interesting about the chemical sense of smell specifically is that signal gets sent to the frontal lobe of your brain, part of your cerebrum, to be identified, but it also gets sent to the emotional pathway, which is part of the limbic system in our brain. So if you've ever smelled something and it just totally takes you down memory lane, that is something that's unique to our sense of smell and the way that it is integrated and processed and makes that possible. So like maybe you can smell like your grandma's perfume and it just totally reminds you of her, that kind of thing. It's pretty cool. All right, let's talk about taste or gustation. Your taste buds are the sensory organ for taste. So we talked about how your eye was a sensory organ for vision. Taste buds are for taste. And they're mainly housed inside of papillae or the bumps on your tongue. 2 types of cells that are important. Gustatory epithelial cells, which are your taste receptor cells, and then basal epithelial cells. So these are actually stem cells that make new gustatory epithelial cells, like when you take a bite of pizza roll and the inside explodes on your tongue and burns the entire thing because you just can't wait to eat it. Then we got to remake those gustatory epithelial cells. So the basal ones are the stem cells that do that. Fun fact, we have around 10,000 taste buds that take in our sensory information for taste every time you take a bite of food. But I say around because everyone's actually born with a different amount, which is why some people are better at distinguishing taste than other people, which is kind of interesting. So how does taste work? I'm going to majorly simplify this, but essentially your sensory receptor cells, those taste receptive cells, They take in the chemical molecules dissolved in your saliva. They send action potentials, essentially down your facial nerve, your glossopharyngeal nerve, and your vagus nerve, which we talked about at the end of Concept 1 for honor students. And then that signal gets sent to the gustatory cortex of your brain, which is going to process it and integrate it and tell your body to release digestive enzymes to break down that food that you took in and get the energy from it that we need. All right, last special sense we're going to talk about, or special senses, we're going to talk about our hearing and balance. And the ear is the special sensory organ that does both of those things, hearing and balance or equilibrium. So we're going to dive into the structure of the ear in the detail that we did the eye. So make sure you're labeling that big body diagram as we go. Our ears are designed to receive mechanical waves, okay, and convert them into nerve signals that our brains can interpret. So hopefully you remember from physical science course that sound creates vibrations, okay, which is the mechanical sound waves or mechanical waves in your ear. And essentially those are going to hit your eardrums, which we can see right here in the picture, and cause these tiny bones in your ear, right here, called the auditory ossicles, to move the internal fluid that's in your ear against a membrane, which will then eventually initiate a signal. So that's like the simplest way I can summarize how it works, but we're going to actually follow this specific pathway now. So it's going to go through here, and that's how it's going to happen. We got your eardrum. Those are your auditory ossicles. And then that triggers these tiny hair-like cells that simulate nerve cells to send the action potentials to the brain. So let's talk through the structure of the ear and kind of how those vibrations travel through it. So 3 general parts. You have your outer or your external part of your ear, your middle ear, which is especially important for hearing, and then your inner ear, which is important for both hearing and maintaining your equilibrium for balance. So let's start with the external or outer ear. A couple of parts. We've got the pinna or the auricle. This is the part we see, which is made of cartilage that you may have a bunch of piercings on. We also have the external acoustic meatus, which is just your auditory canal. That's kind of usually where you're probably sticking a Q-tip to clean some earwax out. The function of our outer ear is to catch sound waves and then pass them deeper into the ear through the auditory canal, which we see pictured here. So we're just going to send them this way. Eventually, it's going to collide with the tympanic membrane, which is just a fancy way of saying your eardrum. And your eardrum is just connective tissue boundary between your external ear or your outer ear and your middle ear. So that's just all that is right there. All right, let's talk about your middle ear. This is also known as your tympanic cavity because it's just behind the tympanic membrane or your eardrum. It's the relay station between the outer and inner ear. And it has 3 tiny bones in it, or your auditory ossicles, which I mentioned before. The malleus or the hammer, the incus or the anvil, and the stapes or the stirrup bone. And these work together to amplify sound waves. We want to make them stronger for when they get to the inner ear because they're going to be moving from air and now into fluid. All right, so we want to make them stronger. And one thing that's fascinating, since we already covered movement in joints in Unit 2, you have tiny synovial joints and ligaments within these ossicles, as well as teeny tiny skeletal muscles that are contracting reflexively to limit the vibration that's going down in these ossicles when a sound is super loud. So this helps minimize damage to your hearing receptors, which is pretty cool how some of those things we talked about in Unit 2 can work on such a teeny tiny level. And then another, so these auditory ossicles conduct vibrations to the oval window to get inner ear fluid moving. So we can see that right there, that oval window. And then you also have the eustachian tube, which I'm just going to show right here, which is just a passageway from the middle of the ear to the pharynx that aids in equalizing pressure around your eardrum. And then it also helps to remove any debris or secretions that may exist in the middle ear. All right. And then we have your inner ear or your labyrinth. And there's kind of 2 parts of this, your bony labyrinth and the membranous labyrinth. So Both divisions are filled with fluid that help conduct the sound vibrations that allow us to hear and then also respond to changes in our equilibrium. Their function is to turn physical vibrations into electrical impulses, so action potentials. So again, we're taking mechanical energy or mechanical waves, we're converting them to electrical. The bony division of the labyrinth is just a system of channels through the bone. The membranous division is just a series of membranous sacs and ducts. Both have a bunch of fluid in them that are conducting the sound. So let's talk more details. 3 parts of the bony labyrinth. So we have your vestibule, which pointed out right here, is the key structure for maintaining your balance. Head movements cause fluid to stimulate hair cells in your ear to then send action potentials through that vestibular nerve, which is pictured here, so that we can respond to that. Your semicircular canals aid in maintaining balance when your head rotates. You can see those pictured here. And then your cochlea, kind of looks like a snail, has a ton of hair cells, and these vibrate at different frequencies. And so they stimulate the organ of Corti to send action potentials through the cochlear nerve, which is pictured there, to the auditory cortex in your brain. Think of the organ of Corti like the microphone of your inner ear. It's just inside your cochlea. So it's just going to magnify that so that then we can send it to your brain. And that is how we hear. Those are our special senses.